speaker is Dr. Gayatri Balasurya, who is a postdoc research fellow at the Kobe University School of Medicine. And she'll be talking about GI dysfunction in dup15q that we also know will be applicable to the other disorders in this room as well. So thank you so much.
Yeah, so thank you very much for the opportunity to talk to you today. So today I will talk to you about the gastrointestinal dysfunction in 15q duplication syndrome and what we know about it and how we plan to investigate it. So the 15q duplication syndrome is a neurodevelopmental disorder, and the exact prevalence in the general population is not known, but it's estimated to be about 1 in 5,000. However, amongst patients refers for diagnosis due to developmental concerns, it is estimated to be about 1 in 500. And many patients with 15q duplication exhibit ASD-related phenotype. The characteristics of 15q duplication syndrome involves hypertonia, motor delays, epilepsy, ASD traits, as well as gastrointestinal symptoms, which is the focus of my study. The main gastrointestinal symptoms seen in the 15q duplication syndrome includes constipation and gastroesophageal reflux. The other symptoms include diarrhea, abdominal pain, bloating, and nausea. There are two interesting studies, one from 2015, which includes 46 patients, and they reported GI symptoms in 76.7 isodysentery and 87.5 interstitial duplication, and no clear association was seen between GI issues and ASD. They reported constipation in 60% of the patients they looked at and gastroesophageal reflux in 56.7% of them. Importantly, they noted that when the GI symptoms were treated, the irritability and aggressiveness improved. The second study is a more recent one. It's from this year, and it included 101 patients. They also reported GI symptoms in 87% of the patients, and again, the constipation was the main GI dysfunction, which was seen in 55% of them. They also reported that GI symptoms occurred prior to the diagnosis of the syndrome in 49.5% of them. A correlation between the challenging behavior and GI symptoms were also reported. Other than these two studies looking at the GI symptoms in the syndrome, not many literature is available, and there are many unanswered questions, such as frequency of GI issues, whether they're seen more frequently during early stages of life when they're younger and less when they get older, whether they have altering phases of diarrhea and constipation, and also whether the dietary interventions such as probiotics, etc., has any beneficial effect. So this highlights the need for further investigation in this field. The gut functions and the gastrointestinal tract is controlled by both extrinsic and intrinsic innervation into the gut. The extrinsic control is through the vagal, sacral, and spinal nerves, and the intrinsic control is through the enteric nervous system, which is the gut's own nervous system. It contains more than 100 million neurons, and these enteric nerve cells, and there are also about 400 million glial cells, and these cells cluster into small groups known as ganglia. And these ganglia are interconnected to form a plexus. So there are two such plexuses embedded within the walls of the gut. The first one is the submucosal plexus, which is embedded between the mucosa and the circular muscle layer. And then there's the myenteric plexus that is present between the circular muscle layer and longitudinal muscle layer. Myenteric plexus is mainly responsible for regulating gut motility, and submucosal plexus governs secretion, absorption, and mucosal barrier function. The neurons of the enteric nervous system have similar neurochemistry to the central nervous system. Acetylcholine is the main excitatory excitatory neurotransmitter of the gut, and nitric oxide is the main inhibitory neurotransmitter. There are many other neurotransmitters such as 5-HT, serotonin, GABA, glutamate, somatostatins, and so on. I will briefly introduce the mouse model that I'm going to use. So in Professor Toru Takumi's lab where I work, there are a few different mouse models related to 15q duplication. One of them is the paternal 15q 11 to 13 duplication model. And this model has shown many autism-related behaviors, including poor social interaction, behavioral inflexibility, abnormal ultrasonic vocalizations, and correlates of anxiety. When they looked at the different areas of the brain, they saw altered 5-HT and GABA functioning and receptor expression levels. A recent study that was published last year showed decreased microbial species diversity in the fecal matter. So what's important about this model is that the GABA cluster, GABA receptor subunit related genes are duplicated in this model, and those GABA receptors are also expressed in the enteric nervous system. And both 5-HT and GABA are important neurotransmitters in the gut. So I'm going to start looking at this paternal duplication model. But the lab also has a maternal duplication model as well as a GABA cluster deletion model. So that leads to the main focus of the project that I'm looking at, to characterize the gastrointestinal phenotype in 15q duplication mouse model in order to identify therapeutic targets to treat GI dysfunction. I started off by looking at the total gastrointestinal transit. To do that, I gavaged this I started looking at the total GI transit. So I gavaged the animals with a carmine red dye and then waited for the appearance of red pellets in the feces. And then I noted the time taken for the process. So in 15q duplication animals, they took significantly longer time for this process, indicating a much slower gastrointestinal transit. And I also started looking at the small intestinal transit where I gavaged the animals with the same red dye and sacrificed them after 10 minutes and observed the dye front within the small intestine. So this research is still ongoing, and there are more to add into this data set. Then I also looked at the arrangement of neurons within the enteric nervous system by performing immunofluorescence using whole mount preparations. So I used HuCD, which is a pan-neuronal marker, and also antibodies targeting GABA and neuronal nitric oxide synthase. So the preliminary results so far indicated there's no significant difference in the number of neurons per ganglion or the percentage of GABA expressing or neuronal nitric oxide synthase expressing neurons. I'm also looking at the percentage of 5-HT and choline acetyltransferase expressing neurons. And this part of the research is also still ongoing. I also set up a video imaging technique in the lab to study the colonic motility ex vivo. So the most interesting about the gut is because it has its own nervous system. When you isolate a piece of gut and cannulate it in an organ bath, as long as you provide the physiological conditions, they can function by itself. So what we do is we video record this contraction, the spontaneous contraction that are seen, and then use an enhanced software to convert these videos into corresponding spatiotemporal maps. The software assigns a warmer color, red-orange color, to the constricted colon and a cooler color to the dilated colon. So over a period of time, in a spatial temporal map like this, the diameter of the colon is plotted against the length of the tissue as a function of time. So in this spatial temporal map, each of these lines will represent a colonic motor complex that originate at the oral end of the tissue and travels all the way to the anal end of the tissue. So from these maps, we can generate a lot of information about colonic motility, because in this case, I've used a mouse colon here. So we can calculate the frequency of these contractions, the length and the velocity of them, as well as the resting colonic diameter. The experimental protocol has 3 main steps: a control period, a drug exposure period, and a washout period, each lasting for 1 hour and consisting of 4 15 minutes recording. So this techniques allows us to compare the colonic motility between 2 groups. So under the control conditions, the gut tissue is exposed to physiological saline, so we can compare changes to this rhythmic activity between the wild type and the 15q duplication model. And during the drug exposure, we can use specific antagonist or agonist to the enteric neurons and study the changes to the colonic motility. So in this case, I've used pituitary, which is a GABA A receptor antagonist. And you can see this is a wild type preparation. And when you add pituitary, the rhythmic activity has disappeared, or it has changed. So we can compare these changes between the 2 groups. This method also allows us to study the restorative effects of different drugs to treat any dysmotility that's seen. So in summary, around 80% of 15q duplication patients exhibit GI dysfunction, and constipation and gastroesophageal reflux are the most common ones. There are several mouse models of 15q duplication available, and paternal duplication of 15q 11.2 to 13 region model shows ASD-related phenotype, altered 5-HT and GABA functioning, and fecal microbial dysbiosis. Therefore, I started looking at this model, and the preliminary results so far indicated an altered total gastrointestinal transit. And this research is still ongoing. So I would value your feedback on this. Yeah, with that, I will conclude my presentation.
Thank you, Dr. Balasuria. I'm just going to pull up some questions. And if there's any in the audience, we can grab a mic for you too. Well, can we get the slides for this session? Yeah, and I think that what they meant is probably a copy of the slides. So if you don't mind maybe sending that to us, we can
I will
disperse. Thank you.
That's okay. Thank you so much.
Great. And the second question is, how does needing a movement break differ from hyperactivity and from ADHD? My 6-year-old IDIC dup15q has trouble focusing on school tasks for more than 5 minutes, but can sit and watch music videos for an hour. As a parent, I can relate to that. She has sensory needs too and has been diagnosed with ADHD. Is medication the way to go, or should we try other methods?
Sorry, I think this question probably meant for the previous.
I think so too. Yeah, I know. It's like in the wrong box.
Yeah.
I will. I'll send it to Dr. Carey. What this one is for you though. What are typical symptoms and indications of GI reflux in younger children who may not be able to explain what they're feeling due to age and developmental delays?
So I'm not a clinician, so I won't be able to give any expert advice on this. So, but all I can say is these research that I reported before are done through questionnaires for the parents or the caretakers. So there's a new questionnaire that's based, it addresses the nonverbal ability of these patients. So it's for the carers, but other than that, I can't, like I'm not a clinician to give a specific answer for that. That could be sent to the previous speaker as well, I guess.
Be for the next panel. So it's perfect timing for us to say thank you so much for your time, and we'll move on to the next session.